First of all, thank you to the organizers for inviting me to speak here today. I'm going to talk about mechanisms of resistance, mainly resistance and a bit of sensitivity to PARP inhibitors in ovarian cancer. So I'm actually surprised this slide hasn't been shown before, but why we're interested in high-grade serous ovarian cancer is because up to 50% of high-grade serous ovarian cancers are predicted to have a defect in HR, homologous recombination pathway, which is a DNA repair pathway, which could result in DNA, HR deficiency. And these could include germline BRCA1 or BRCA2 mutations, but also somatic BRCA mutations, and also, as mentioned before, BRCA1 methylation, as well as mutations in other HR-related genes. So RAD51C or PALB2 and so on. And so, again, a reminder why PARP inhibitors is a targeted drug in high-grade serous ovarian cancer is because it works via a mechanism of synthetic lethality, where cells, upon PARP inhibition, will, with HR deficiency, will succumb to cell death, while cells that are proficient in HR should survive. And so it makes it an attractive targeted treatment for high-grade serous ovarian cancer. And although there's, we know that PARP inhibitors work in HR-deficient cancers, we also know that resistance often occurs. And there are two different types of mechanisms of resistance, pre-existing and acquired. And in this instance, I'm talking about pre-existing resistance where it would arise either during previous chemotherapy rounds or before at the time where tumor was chemo-naive. And there are multiple mechanisms of PARP inhibitor resistance that have been described in literature to date. Some have been described in vitro, some in vivo, and some in the clinic. And these include restoration of HR pathway, so secondary mutations or reversions, also alternative isoform expression, structural reversions. Also not mentioned today, a loss of another DNA repair pathway, which is a classical non-homologous end joining pathway. This has been documented in vitro. Mutations in PARP1 drug binding site have been proposed as a mechanism of resistance as well, because they would prevent the drug from binding to the site. Oncogene overexpression has been proposed as a mechanism of resistance. And finally, as Liz has talked about, drug efflux, so P-glycoprotein overexpression. So today I'll focus on only two mechanisms of PARP resistance: secondary mutations and alternative BRCA1 isoform. And the work that I'll be presenting today is focused on ARIEL-2 Part 1 trial. And here the investigators performed next-generation sequencing of the tumors to stratify them by HR pathway status. And patients were grouped into three groups: BRCA1 or 2 mutant, then BRCA wild type but LOH high, and LOH in this instance means genome-wide loss of heterozygosity, which is an indirect marker of HR deficiency. And then the third group was BRCA wild type but LOH low group, so likely HR proficient group. And here I'm showing the results of ARIEL-2 Part 1 trial. You can see in blue is the group of BRCA1 or 2 mutant patients, and they performed the best in comparison to BRCA wild type but LOH high group, so HRD positive in a sense here. They performed sort of in the middle. They had an intermediate response, and then BRCA wild type by LOH low group performed the worst. But if you look at the duration of response in the patients with LOH high group, but without BRCA mutation, they performed similarly well to BRCA mutant group. And here I'm showing a swimmer's plot of the responders in ARIEL-2. And you can see that a number of patients had BRCA1 or 2 germline mutations or somatic mutations, but also there were a few patients that had methylation of BRCA1 or RAD51C, as well as mutations in other HR pathways, so RAD51C or NBN here. But also importantly, you can see that although all of these patients did respond at some point, a large majority of these patients developed resistance. So it's important to study the mechanisms of resistance to understand how we can actually overcome it. And we were quite privileged to work on a subset of the tumors collected throughout the trial. We looked at 12 patients where pre-treatment and post-progression biopsies were collected, and Foundation 1 next-generation sequencing was performed on all of these 12 tumors. And you can see the results on the right. Basically, this is just summarizing the mutations and copy number changes that were detected in these patients. And I'm not sure if you can read it, but there are 3 bars basically for 3 or 2 bars for each patient, and the bar in the blue is the post-progression biopsy, while the 2 left bars are the archival or pre-treatment biopsy. And in 12 of these patients, we found 6 patients that had HR defects in their primary cancer. And surprisingly, 5 of 6 post-progression biopsies that were sequenced had a secondary mutation in the HR gene. The only one that didn't have a secondary mutation was one of the case 2, which had BRCA1 mutation. So I'm going to talk about 3 cases today. One case with BRCA1 mutation, one with RAD51C, and one with RAD51D. So first of all, secondary mutations, they were mentioned by Liz and Ursula, but secondary mutations in BRCA1 or 2 have been previously described in literature as a mechanism of PARP inhibitor and platinum resistance, and it's the most described mechanism of resistance to PARP inhibitors to date. However, they haven't really been described in other genes so much, in other HR genes. So in the case 1, we had a primary mutation, which was a frameshift insertion, which caused premature protein termination of BRCA1. And then in the post-progression biopsy, we detected 2 secondary mutations. One of them was a sort of a classical secondary mutation where there was a deletion before the primary mutation, which restored the reading frame of the protein. And the second mutation was actually a splice site downstream of the primary mutation, and this splice site caused this delta 11q isoform, which is a known isoform of BRCA1, and it's a shorter, partially functional isoform. And this isoform lacks a large proportion of exon 10, where the primary mutation was sitting. So in a sense, this splice site mutation spliced out the primary mutation, so you ended up with the shorter but partially functioning BRCA1 isoform. And interestingly, this BRCA1 delta 11q has been previously reported as a mechanism of PARP inhibitor and also cisplatin resistance. So this finding sort of supports this as a mechanism of resistance further. Now, in the second case, we looked at a patient with germline RAD51C mutation. And this patient had 1 prior regimen of chemotherapy before entering into the trial. And throughout the trial, 3 lesions were monitored, all of which had partial response. And after 11 months of treatment, this patient developed a newly enlarged groin lymph node, which upon progression was biopsied, and we detected 4 secondary mutations within this 1 lymph node. So we wanted to study these secondary mutations to understand their clinical importance. First of all, to recap what mutations we detected, in the normal tissue, germline tissue of the patient, we saw 1 copy with RAD51C nonsense mutation, which caused premature termination of the protein, while the other copy was wild type. Then in the pre-treatment biopsy, we saw a loss of heterozygosity event. So we only saw this RAD51C nonsense mutation. And then in the post-progression biopsy, we detected 5 different events. So the primary mutation, alongside with the 4 secondary mutations, all of which sat on top of the primary mutation and restored the reading frame of the protein and restored the full length of the protein. So, as I mentioned before, we wanted to model these to understand what they did exactly. First of all, we performed some deeper sequencing of this lymph node. So we had 2 cores that were collected, and we were able to sequence them independently. In fact, the second core, we sequenced 2 different sections of the second cores were sequenced separately, and we saw an incredibly high degree of heterogeneity of these secondary mutations. So in the first core, we saw all of the 4 secondary mutations, while in the second core, we saw only 2 of the mutations present. And this is within 1 lymph node. So the degree of heterogeneity is crazy. We then also performed serial sectioning of the leftover material of this second core to see the degree of heterogeneity even further. So we were able to see a subtle shift in the allele frequency of these secondary mutations here. We also modeled these secondary mutations in vitro in a ovarian cancer cell line model, so OVCA8 cells. We first knocked out RAD51C in these cells and then reintroduced different transcripts with either primary mutation or secondary mutations. And as expected, we saw that the primary mutation and the knockout cells had sensitivity to PARP inhibitors and platinum agents, while the secondary mutations behaved similarly to the wild type RAD51C. So they restored the function of HR. And we confirmed that by performing RAD51 foci assay, which is an indirect marker of HR proficiency. And also we saw similar results with the HR reporter assay, which is a more direct measure of HR pathway activity. So again, with the primary mutation, we saw lack of foci formation and also no HR activity, while secondary mutations caused reversion. And the third case that I'll be talking about is a case with a germline RAD51D mutation. So this patient had 2 prior regimens of chemotherapy before entering onto the trial. In this patient, also 3 lesions were monitored, 2 of which were responding and 1 was never responding throughout the trial. And upon progression, this splenic non-responding lesion was resected and a secondary mutation was identified, while in the responding lesion, which was resected at the same time, only the primary mutation was detected. So again, we wanted to model this, and we performed in silico modeling of RAD51D protein. We couldn't, unfortunately... Oh, it started. Okay. So here I'm showing a simulation of 6 wild-type RAD51D molecules wrapped around double-stranded DNA. And as we zoom in, you can see in the red residue is the bit that gets substituted in the secondary mutation. And you can see that that area seems to be interacting with double-stranded DNA. So we wondered whether that be retained in the secondary mutation. And we performed modeling of the secondary mutation, and yes, we saw that there was some interaction with double-stranded DNA still retained, which has suggested that the secondary mutation may have retained most of the protein function of RAD51D. Again, we wanted to confirm this in vitro, and we performed this in PO4 cell line, which is a high-grade serous ovarian cancer cell line. And we knocked out RAD51D in this case, and the knockout was quite a similar mutation to the primary mutation. And then we also introduced secondary mutation by CRISPR technology. So here, as expected, we saw that the knockout resulted in rucaparib and cisplatin sensitivity, while secondary mutation resulted in resistance. We also performed some RAD51 foci assay, but you can't really see it on the projector, I think. But basically, we saw similar results where secondary mutation caused restoration of HR pathway, while knockout did not have active HR pathway. So to summarize, we saw 5 of 6 patients that had HR DNA repair pathway defected in their primary biopsy had secondary mutations upon relapse. So these were in RAD51C, RAD51D, and BRCA1. We identified a secondary splice site, BRCA1 mutation, as a Delta 11q isoform, which results in resistance to PARP inhibitors. We also confirmed that RAD51C and D are in fact HR genes, and mutations in these result in PARP sensitivity, while secondary mutations result in resistance. But there are still questions that need to be answered. So how common are these mutations in PARP-treated patients? And hopefully future trials will be able to answer this. But we do need to collect post-progression biopsies or circulating tumor DNA at least to actually answer this. And then also the question of pre-existing versus acquired. And this is important to determine the best sequence of treatment for the patients. So in the RAD51D case that I showed you, one of the lesions was never responding throughout the trial. So it's likely that this mutation was pre-existing and possibly arose during the two previous rounds of chemotherapy. While in RAD51C case, where a lymph node was enlarged only after 11 months on PARP treatment, and 4 different mutations were detected in this lymph node, it's likely that this is an acquired mechanism of resistance here. And so on this note, I'd like to thank everyone involved in the project, specifically Clare Scott, who is the lab head, and also Clovis Oncology, who we did this work together with, and Liz Swisher and Maribel for helping out with sequencing. Thank you.